Good afternoon, everyone, and welcome wherever you are around the world to this webinar hosted by the IUIS. It's a really exciting webinar for me to be hosting. My name is Professor Johnny Peter. I'm from the University of Cape Town in South Africa. Behind me, you can see the mountain, which hopefully will also stimulate you to think about IUIS Congress coming eventually in Cape Town, and we're excited to welcome you all there. But in today's session, we're going to be hearing about immune responses to SARS-CoV-2. Akiko Iwasaki, she's going to discuss the impact of mutations, of variants on vaccine-induced immunity, looking at key adaptive immune players in the clearance of primary infection and also prevention of infection by vaccines. Talking briefly as well about new vaccine strategies to elicit mucosal immunity, to block infection and disease, and then also touching on long COVID and her work there. I don't think really Akiko Iwasaki needs much introduction in the immunology world. And for those of you, what I was actually mentioning just earlier before the webinar started, is that the Twitter world has brought us all close. And I'm certainly an avid follower on Twitter of Akiko Iwasaki's lab's work, very enviously looking at the long list of authors that she's able to rally to get this fantastic science that she does up and running. But she is a Sterling Professor of Immunobiology and Molecular, Cellular and Developmental Biology at Yale University, as well as an investigator of Howard Hughes Medical Institute. She was a PhD from University of Toronto with a postdoctoral training in NIH. And her research has focused on the mechanisms of immune defense against viruses at mucosal surfaces. And she is leading the Yale COVID-19 recovery study, which is really focused on this particular area that she'll cover today, as well as leading many other investigators focused on multiple pathobiology of long COVID. So I'm going to hand it over to you. Just also, while the speaker is talking, please post your questions. We're going to have a good question and answer session. So there's a Q&A. Please post your questions there, and we'll address them at the end of the talk. I'm going to hand over to you, Akiko. Thank you.
Thank you very much, Professor Peter, for the kind introduction. And I'm very happy to be here today in this seminar. And hello to everyone. I see the public chat with names and people from many countries. So hello and welcome. So I'd like to talk about our recent work on immune responses to SARS-CoV-2. And, I will leave plenty of time at the end for lots of Q&A. So I do encourage you to post your questions on the Q&A. So today what I'd like to do is to give you two separate stories. One has to do with longitudinal immune responses that we've monitored in patients who had COVID. And we found some interesting differences in the immune differences that may explain the disease outcome differences. And then in the second half, I'd like to talk about what we know and what we don't know, which we don't know a lot, but we are learning a little bit about what is long COVID and the possible disease pathogenesis there. So as Professor Peter mentioned, I have been very fortunate to rally a large number of collaborators to tackle immune responses to COVID. And this was made possible by an establishment of a biorepository called Impact Yale, which was initiated by a large number of people, including Dr. Albert Koh at the School of Public Health, who is the head of this repository. And what we were able to do is to collect specimens from participants in three different cohorts. The first cohort being the inpatients who were in the Yale New Haven Hospital. And from these participants, we were able to collect a variety of specimens, including blood, saliva, nasopharyngeal, oropharyngeal, and nasal rectal swab, as well as feces and urine over time. And then from the second cohort of patient, which are the outpatients, we were also able to collect information about their symptoms as well as some biospecimen over time. And then the third cohort of participants were the healthcare workers. And these healthcare workers were providing a very important specimen as controls, as well as we were able to administer daily testing from saliva and NP swabs. And this was very important in the beginning of the pandemic when testing was very, very inaccessible. And we were able to do this, provide this service for the healthcare workers on a daily basis. So using this IMPACT-DL study, we were able to monitor longitudinal immune phenotyping of COVID patients. And this work was led by 4 brilliant trainees, John Klein, Patrick Wong, Carolina Lucas, and Tiago Castro. And of course, along with a number of collaborators. And so what we did in this study was to monitor immune responses in COVID patients who were hospitalized in the Yale New Haven Hospital. And we divided the patients into 2 large categories: moderate patients in the hospital floor and severe patients in the ICU. And the moderate and severe patients were further divided into 3 different clinical scores. 1, 2, and 3 are divided according to the supplemental oxygen requirement that these patients had. As you can see, none, 1 to 3 liter per minute, or 2 or 3 or more liter per minute. And then 4, 5, and 6 refer to patients who were in the ICU with supplemental oxygen of 6 liter per minute or more, or mechanical ventilation, or 6 being death. And then we had the uninfected healthcare worker controls. So this was one of the first panels that I was able to see from the real-time analysis of PCR from the PBMC of these participants. And the green dots indicate healthcare workers. The blue dots are the moderate patients, and the pink dots are the severe patients. And the first striking thing that we noticed was that the T cell numbers drop quite considerably in both the moderate and severe patients. And this had already been reported at the time from our colleagues in China. But this appears to be a significant feature of COVID, is that the circulating T cells drop in number, as well as we don't think this is a sequestration issue in other tissues, because we also don't see accumulation of a large number of T cells in other organs either. So there's something going on with the T cell numbers and potentially lymphogenesis. The other features that are obvious is the increase in monocytes in disease severity, as well as neutrophils and eosinophils that have that come into the PBMC fraction as a result of their low density status. And so this is also interesting because these types of cells are usually excluded from PBMC, but perhaps due to their dysfunctional or immature nature, they end up being in the PBMC fraction in moderate and severe patients. We also see the many cytokines and chemokines that are elevated in patients, and in particular, things like IL-1, IL-6, TNF, interferon. These are things that we can readily measure in circulation. And so what appears to distinguish the severe versus the moderate patients is the nasopharyngeal viral load that we can detect over time. So on the x-axis is the days from symptom onset, and on the y-axis is the logarithmic scale NP viral load, RNA viral load. And you can see that the severe patients have high levels of NP viral load over time that does not decline, whereas moderate patients do ultimately reduce their viral load to below the threshold. So this appears to be a feature, and we also see this in the saliva as well. And what appears to be going on here is that it's the viral load that's driving antiviral cytokine levels, and not the other way around. So if you look at the x-axis, which is the viral load in the nasopharynx, and y-axis being the different types of cytokines we measure from the plasma, you can see that there is a positive correlation between the viral load and interferon alpha and interferon gamma. And the other way to interpret this data is that the higher levels of interferon is not leading to lower levels of viral load. So this suggests the capability of the SARS-CoV-2 to evade from interferon-induced antiviral effector mechanisms. That's been shown by multiple other papers. And then, of course, other inflammatory cytokines like TNF and TRAIL are also elevated in accordance with the viral load. And when we looked at other cell types and other features that can be found within the peripheral blood, what we see in column D are the type 1 immune cells and cytokines such as monocytes, IL-12, and interferon gamma. This is the typical antiviral effector that we expect to see in a acute respiratory viral infection. However, in column E, we also see type 2 immune responses such as eosinophils, IgE, and IL-4, IL-5, IL-13, these typical type 2 inflammatory cytokines being elevated and maintained at high levels in the severe patients compared to the moderate patients. And this was a bit strange when we first saw this, because we don't often associate these type 2 immune effectors with a viral infection. And type 3 immune responses that we often think of for extracellular pathogens, such as neutrophils, IL-6, IL-17, and so on, are also being elevated in the severe COVID patients over the moderate patients. So when we took the plasma factors and did an unsupervised clustering analysis, what we found were 3 clusters of patients based on the plasma levels of these factors. So each column here is a patient, and each row is a factor that we measure from the plasma. And you can see that the first clusters of patient in the middle has the highest levels of tissue repair growth factors, such as EGF, VEGF, PDGF, IL-7, and so on. The second cluster of patients on the right side of the panel here it has, in addition, chemokines and mixed cytokines being elevated. So the darker the purple, the more factor there is in that plasma. And the third cluster of patients, which is the leftmost columns, you can see that these patients have very high levels of type 2 and 3, as well as mixed cytokines in their plasma. And what was very interesting is that when we follow these patients over time, the first cluster of patients who had the tissue repair growth factors were the ones that ultimately controlled the clinical score and were discharged alive from the hospital. Whereas the second and third cluster of patients have increasingly worsening clinical score and had the highest frequency of coagulopathy as well as mortality. And when we took all these factors and cell types and asked for biomarkers for COVID-19 mortality, what we saw is that the top biomarker turned out to be IL-18. So it's at the very bottom of this graph. You can see that IL-18 has the highest association for mortality with COVID. So this was informative in thinking about the pathogenesis of this infection. because it appears that the IL-18, which is a factor released as a result of inflammasome activation, is the most associated risk factor for mortality, and then followed by interferon alpha itself. So this interferon alpha, as I mentioned, correlates with the viral load, and it's indicative of the failure to control the viral load in the initial setting of the infection, and that this late phase interferon is actually being counterproductive and potentially immunopathological. And this is followed by other factors like IL-10 and neutrophils and so on. In a separate study we did with a computer scientist, Dr. Smita Krishnaswamy, they found that the most associated cell type that we can find in circulation for mortality is indeed neutrophils. So if you put all this together, it appears that the neutrophils and IL-18 secretion and this inflammasome activation within these myeloid cells may be the driver of these types of lethal diseases. So what about adaptive immune response? So we took a look at the timing of the antibody response and wanted to correlate that with the COVID outcomes. And this study was led by, again, Carolina Lucas and John Klein. And in this study, what we did was to monitor the levels of antiviral antibodies in circulation. And here we also included mild cases, outpatient samples as well. So just to take you through this graph, the left two panels indicate anti-spike antibody IgM and IgG, and the right panels indicate the IgM and IgG against the receptor binding domain of the spike protein. And the negative are the healthcare workers who were never infected with SARS-CoV-2, and you can see that their levels are quite low. And then the non-hospitalized is the next sort of lighter purple color, which indicates that the mild cases generated lower levels of antibodies to spike and RBD. And then the next three are the moderate, severe, and deceased. These are the hospitalized patients. And you can see that the hospitalized patients have much higher levels of antibody against the spike and RBD. And even those patients who died of this infection in the lightest purple, they did generate a significant level of antibody against the spike and RBD. So if you just look at the maximum level of antibodies alone, it's very difficult to predict the disease outcome of these patients. However, when we looked at the timing of these antibody responses, there we saw something significantly different. For instance, here indicated in purple are the deceased patients, and IgM and IgG against the spike and RBD are again measured. And you see that these patients tend to have much delayed onset of antibody generated against the spike and RBD. Compared to the discharged patients, these are people who were discharged alive from the hospital in green. You see that they mount a rapid response to the RBD and spike. We also found a handful of patients whom we are calling the high neutralizer, or HN. They had high levels of neutralizing antibodies from the very first sampling, and they tended to do better with the disease outcome. And so these patients had very high levels of antibodies against RBD and spike throughout the course of disease. And when we divided the patients into two groups, the early neutralizers who made the neutralizing antibodies prior to 14 days of symptom onset versus the late neutralizers who did so after the 14 days of symptom onset. And we followed disease course over time. On the right panel, you can see that there's a divergence of disease course in patients who are late neutralizers, which is the green line. You see that they have worse disease outcome at later time points of collection, whereas the early neutralizers, their clinical scores ultimately come down. So again, emphasizing that this delayed onset of antibodies to the spike protein and neutralization capacity can divide the patients into those who recover versus those who develop worse disease outcome. So this first part of the talk, I showed some evidence for persistent viral load in the severe disease patients, and that this viral load correlates with the antiviral and inflammatory cytokines. And we also showed in that paper that increased interferon and inflammatory cytokines within the 12 days of symptom onset were predictive of mortality. And there are signatures in the plasma proteins that can distinguish patients into those who recover versus those who develop worse disease. And in patients who develop worse disease, the worst diseases are the type 1, 2, 3 cytokine mixed signatures. And we consider this to be a maladaptive response to a viral infection. And finally, we show that the delayed onset of neutralizing antibodies also correlate with mortality. So now that we have been studying acute COVID immune response, I'd like to turn our attention to the long COVID. And this is a very important aspect of this pandemic, that there's a parallel pandemic that's happening with acute and long COVID. And long COVID is happening in estimated to be 10 to 30% of the survivors of COVID. And this accompanies numerous symptoms that are debilitating to patients. And so there's an urgent need to study the pathobiology of long COVID. So there are at least 3 paths to long COVID. And just to emphasize that it's not just the severe and critical COVID patients that become have this persistent symptoms for a long time, but also mild and moderate disease. And in some cases, asymptomatic SARS-CoV-2 infection can lead to a number of unexplained new onset symptoms that include fatigue, post-exertional malaise, and many neurocognitive deficits, as well as respiratory, cardiac, and musculoskeletal issues. And so there are over 200 symptoms reported for long COVID. And this is just a handful of common ones, but it's not an exhaustive list, just to indicate that there are pretty much all the organ systems that we can imagine are involved in the symptom features that are reported, including systemic ones as well as organ-specific deficit. And some people have one of these, and while others suffer from multiple. Some patients have more than a dozen of these symptoms ongoing at the same time. And this is really explains the debilitating nature of this disease. So how are these long COVID disease, what is the pathogenesis? And currently, we don't really understand how this viral disease causes these numerous other symptoms in multiple organs, but there are some hypotheses, and these are the main ones that I listed, but again, it's not an exhaustive list. First possibility is that there may be viral reservoir or viral PAMPs, pathogen-associated molecular pattern, that may be remaining in these patients, and that may be driving both innate and adaptive chronic inflammation. And on the left of that, I listed some papers that support such a hypothesis, such as the detection of viral proteins and viral nucleic acids detected from intestine, lymph node, and many other organs throughout the body in people who are infected with COVID, infected with SARS-CoV-2. The second hypothesis is autoimmunity. There may be T and B cell responses to a variety of tissue-specific or cell type-specific antigens that may be driving these numerous symptoms. And on the right, I gave a couple of examples of papers that have demonstrated that. And the Walcott et al. Paper, for instance, found that all 31 participants in their study had anti-GPCR autoreactive functional antibodies against G protein coupled receptors. Third hypothesis is dysbiosis of microbiome or virome. And this may be reactivation of latent viruses or dysregulated microbiome in the gut. A paper by Su et al. In Cell recently showed that EBV reactivation at the time of diagnosis was one of the 4 factors that are predictive of long COVID. And finally, there's also tissue damage or distal inflammation that causes dysfunction in other organs. And this is something that I'm going to talk about a little bit. So there are a number of studies now monitoring immune differences in patients with long COVID and who are the matched controls here, who have had COVID but ultimately recovered in blue. So the long COVID patients in red, you can see that they maintain high levels of interferon beta and lambda-1 at 8 months observational time, compared to the matched controls who ultimately have lower levels at 8 months post-infection. And in fact, this study found that the top associated factor for long COVID was indeed interferon beta. So that suggests that there may be at least a viral PAMP or some sort of innate activation that is chronically stimulating cytokines such as interferon in these patients. As I mentioned already, there are autoantibodies to various G protein coupled receptors, as well as the typical lupus-like autoantibodies that are found in patients with long COVID. And this study, as I mentioned before, studied 31 participants with a neurological PASC, and they found that all of them had between 2 and 7 different GPCR-specific functional autoantibodies. And what the implication of this study is that these types of autoantibodies may cause dysfunction in target tissues that are expressing these GPCRs. And another paper that I also already mentioned, the Cell paper in Su et al., they also studied, these are the participants that they studied for up to 2 to 3 months after the diagnosis, and found 4 PASC anticipating risk factors, which included type 2 diabetes, SARS-CoV-2 RNA-emia, Epstein-Barr virus, viremia, and autoantibodies at the time of diagnosis being predictive, the risk factor for long COVID. And here on the right, I also took a panel that I thought was interesting, that they found that negative regulator of circadian rhythm was elevated in neurological symptom reporting PASC patients, as well as cortisol and cortisone being lower in the respiratory viral symptom reporting patients. So there may be symptom-associated changes that's occurring in these long COVID patients. And these types of differences can inform what kind of therapy might be appropriate and suitable for the long COVID patients with distinct symptoms. But we still have a lot of long way to go in terms of really defining the different subsets of long COVID patients, as well as understanding what type of therapy might be most suitable. And this is a study that my team had participated, which is led by Michelle Monge's group at Stanford. And the aim of this study was to look for the consequences in the CNS of respiratory-only mild SARS-CoV-2 infection. And we were able to set this up. So just before I get there are three ways to think about a neurological consequences of this respiratory infection. One is that there may be direct infection that's happening. Second is that there may be autoimmunity that's attacking cells of the nervous system. And third is that there may be an impact of distal inflammation that's happening within the CNS. So we modeled the third possibility in this study by generating an animal model. Actually, we already generated this model before this, but utilize this animal model in which we can introduce the human ACE2, which is the receptor for viral entry receptor for SARS-CoV-2, intratracheally. So this allows this animal to become infected only within the lung, but nowhere else. And after about 14 days, we can infect these animals with SARS-CoV-2 intranasally. And we can confirm that there is infection in the lung of these animals, but not in the brain, because the brain does not have the human ACE2. And that's what's shown in the slice there. There's no SARS-CoV-2 antigen-specific, antigen-positive cells there. And also, this is a very mild infection that none of the animals lost any weight, and we see no sickness behavior exhibited by any of these animals. So it's a very mild respiratory-only infection. And however, we saw a significant changes in the CNS. For instance, on the top panel, we're showing that the white matter of these animals at 7 weeks post-infection had increased reactive microglia, as well as on the middle panel on the right, I'm showing you that there appears to be an impaired hippocampal neurogenesis, which is indicated by the purple signals there at 7 weeks post-infection. And on the bottom, what I'm showing you is the loss of oligodendrocytes. There's about a 1/3 drop in the number of oligodendrocyte, as well as loss of myelination of the axon that we can see in the white matter of these animals. And so this implicates that even a respiratory-only mild infection can have significant impact in the CNS. And in this same study, we demonstrated that there's an elevation of certain cytokines and chemokines in both the circulation as well as within the cerebral spinal fluid of these animals, even at 7 weeks post-infection. So how this is mediated and what kind of changes are undergoing in the CNS is something that we're examining right now. But it could be that some patients with long COVID may be suffering from an event that happened in distal tissue that could still be triggering some neurocognitive deficits. So our overall mission is now to understand immunological and physiological determinants of disease pathogenesis of long COVID. This is, we believe, is the only way to inform rational diagnosis and therapy. And this involves a number of groups, not just Yale, but many groups that we've been able to collaborate with. And it's been supported by the Howard Hughes Medical Institute. So I want to just highlight all the other authors of the studies that I was able to discuss today. And many of these are people from the Yale Impact cohort, as well as many other groups. And I think I have one more slide, which is the most important slide. This is to just acknowledge and thank the members of my laboratory who worked tirelessly to not only conduct the research, but also implement this healthcare worker sort of PCR testing on a daily basis, which often took beyond midnight to run these assays. And the Impact Yale team, without whom none of the translational studies could have been done. And I'm also listing long COVID collaborators here, as well as Mount Sinai, and then microglial collaboration that I was able to describe briefly today, and the funding agencies. So thank you very much for your attention.
Great, thank you very much. Just to encourage people to put their Q&As now. There are a couple of people definitely putting in some good questions, but please feel free while we continue this discussion to pop things in the Q&A. I think like you've done with your talk, Akiko, we'll just try and put the questions for the kind of primary immune response to infection first, and then we go to long COVID next. Maybe a kickoff question, which I saw in the Q&A, which sort of sets up discussing other things, is just your view on what the relative contribution you think from virus-derived cytopathy or sort of secondary immune pathology. I think that's important. Somebody asked that question.
Yeah, an excellent question. So I think that's really the key in understanding this disease, is that there appears to be at least two phases, perhaps three phases if you include long COVID, two phases of during the acute phase, which is that in the very beginning of the infection, the antiviral response is what's an appropriate host immune response. The first time you're exposed to this virus, you really need to mount both quick innate and adaptive immune responses. And when that fails, and then you get into the second phase of this disease, which is mostly driven by the immune response itself. And that type of distinction is also revealed by the usefulness of different therapies. For instance, the antiviral type of therapies work best during the very early phases of this infection. So the earlier, the better. If you give, for instance, antiviral, direct-acting antivirals, or giving monoclonal antibodies or convalescent plasma, they really work very well during the first phase of that response. But when you wait till the second phase, when in sort of hospitalized patients and late phase disease, that's when you really need to modulate the immune response. And that's also consistent with the use of dexamethasone and other immunomodulatory agent during that phase. And that phase, as I mentioned, is really driven by the immune response. In particular, this engagement of the inflammasome response, neutrophils activation, netosis that potentially block vessels, the blood vessels, and other tissue damage that can be induced by that. And so these are the kinds of responses. I think they're engaged because of the failure of earlier control of the virus and the compensatory immune response to try to combat this virus is actually backfiring. And now you're kind of causing damage response. And that's what we also see with the flu. In the severe case of flu, I think that's what's going on, is that this sort of failed attempt at clearing the virus early on is now engaging the inflammasomes and netosis, and that's leading to lethality. So hopefully that kind of paints the picture of what might be happening here.
Sure, and I guess a follow-on question to that is, how do you feel that the viral variants are impacting that variative balance? Because I know certainly here in South Africa, with our last Omicron variant, we were fairly convinced, and I think this is supported by published data now, that the Omicron variant was causing less severe disease. And so one of the things that we've been postulating is, has the mutations in the spike actually impacted on pathogenesis, and maybe potentially the link to the endothelial or vascular injury. But I don't know what your views on that.
Yeah, so that's a very complex question, because you also have to take into account the existing host immune responses in the population. And so I think, the viral intrinsic differences are absolutely there, plus the fact that part of the population was protected because of pre-existing immunity. and obviously public health measures and everything else also is involved. But I think if you just focus on the virus intrinsic property, Omicron is obviously much more transmissible than the other variants. And it also appears to have a distinct tropism. And so, for instance, like you find it mainly in the upper respiratory tract, and then in some cases you go lower down in the tract, whereas the other variants may have been much more capable of going directly to the lower respiratory tract and infecting cells there. And so perhaps this kind of subtle differences in tropism, as well as its ability to replicate in different cell types, potentially different niches, may have led to the difference in the severity, plus obviously the host immune response that's pre-existing or not. And so, it's a complicated question to answer, but I believe that the variants are absolutely impacting the immunopathology as well. And that's something that we really need to investigate. We don't really have a great grasp on what's going on with the Omicron.
Great, and thanks. And the one thing I wondered about was, in the light of the human COVID genetic effort, and I see there's a question here as well from a colleague of mine in Cape Town about whether you looked for autoantibodies to interferons in your Yale cohort.
Yeah, so that's a study that I didn't include due to time, but we did actually investigate autoantibodies to not just cytokines, but human exoproteome. So in collaboration with Professor Aaron Ring's lab here at Yale, who developed a new strategy called Rapid Extracellular Antigen Profiling, or REAP. And REAP is essentially a yeast display library that displays human exoproteome. So anything that's secreted or on the transmembrane, he has now up to 6,000 different exoproteome displayed. And using that technology, we saw a lot of anti-interferon antibody in the severe COVID patients, just like what's been reported from Professor Casanova's laboratory, and as well as numerous other cytokines and chemokines and cell surface of leukocytes. And in some of these patients, we saw that the antibody against the cell surface marker of leukocytes actually was correlating with the depletion of the target cell type. For instance, we saw this in T cells and B cells. So it's almost like there's a different way of having immunodeficiency, not just genetically, but with an antibody, autoantibody. So this kind of interestingly revealed immunodeficiency mediated by autoantibodies, and that appears to be playing a major role, because patients who had very low number of B cells due to these autoantibodies also failed to mount antiviral antibody responses. So there's a wealth of information to be gained by looking for autoantibodies and these functional autoantibodies that may dictate the course of disease. And so that's what we're also doing currently with the long COVID patients.
Great, thank you. And then the other question I wanted to ask you was, maybe close to your background, was you didn't really touch on actual site of disease, mucosal immune responses. Do you think that those are going to be markedly different from what you're seeing in the peripheral blood? And, do you want to make a comment on that as their importance? Because I know a lot of vaccines now are starting to shift to target that approach.
Absolutely. So yeah, I mean, before COVID, we were focusing on mucosal immunity, and we are actually indeed looking at mucosal immunity, and that's why we collect the saliva and nasopharyngeal swabs from all the participants. And we have a preprint that it shows that, for instance, the saliva viral load is a much better correlate of disease outcome than even the nasopharyngeal viral load. So even within these two different mucosa, the oral cavity and the nasal cavity, there appears to be significant difference in the immune response as well as viral, perhaps pathogenesis there. And another sort of follow-up study that we're doing in that space is just to look for immune correlative protection in the oral cavity and in the nasal cavity. And those also appear to be very distinct, that different immune effectors are playing a role. And the plasma levels don't necessarily tell us much about what's going on these mucosal surface. So absolutely, that's an important area of study. And just if I may add one more, since you mentioned vaccines, which I didn't have a chance to talk about today, but we are making a prime and spike vaccine, which kind of leverages the immune effector and memory responses that are created by the conventional vaccine and redirecting it to the nasal mucosa by delivering a booster that is just recombinant spike protein. And this enables robust immune induction in the nasal cavity as well as in the lower respiratory tract, and then secreting IgA, establishing tissue resident memory cells. And these animals are quite resistant to becoming infected again with a SARS-CoV-2, as well as really creating a long-lived immune responses in these tissues. So all in all, yes, we are very interested in mucosal immunity.
Sorry, is that booster, is that given intranasally, or how are the animals vaccinated?
So the prime can be, we believe anything, but the mRNA vaccine, intramuscular, is what we're priming with. We're now testing other types of vaccine platforms to see if that's also compatible with this prime and spike approach. And the important thing is that we give the spike recombinant protein in the absence of any adjuvants, just to avoid any inflammatory responses in the via this route.
Great. Okay, maybe you can then, there are a couple of questions coming through, related to, I guess, some of the more clinical risk factors coming through. So, there was one question here about pathological autoantibodies being related to age. There are also the factors that you mentioned about, sort of the well-known things of type 2 diabetes. And I wondered maybe you can comment on the role those are playing in terms of their drivers of the immune pathogenesis or, in severe COVID.
Yeah, so comorbidities are absolutely an important risk factor. Age is a huge risk factor, and age also drives these comorbidities. So absolutely, there are many sort of host pre-existing host conditions that can drive these severe disease. And the last part, I was also mentioning that autoantibodies that are typically found for lupus-like autoantibodies, was one of the sort of 4 predictive factors for developing long COVID, as well as type 2 diabetes in that case. So, but these were mostly hospitalized patients in that study. And we don't know whether the same kind of risk factors apply for developing long COVID in the mild disease cases. And that's what we're currently trying to figure out. But yeah, so if you can think of the host, as having multiple parameters that can predispose to both severe disease and long COVID, and they might be different. But age, sex, comorbidities, these are definitely some of the drivers of severe COVID. And interestingly, which I didn't emphasize for the long COVID, the age and sex are important, but in the other way. So for the severe COVID, we see that the older age and male sex is more associated with severe COVID outcome. Whereas in the long COVID after the mild disease, it's really women in their 20s to 60s. These are the sort of demographic that's really enriched in the long COVID after a mild disease. So there again, like you see immediately that there are different risk factors that's driving these 2 outcomes. And the latter group, the women in their 20 to 60-year-old, this is also a group that's most highly susceptible to a variety of autoimmune diseases. So there may be different risks that's associated, that are underlying these 2 outcomes.
And maybe specifically, to sort of aspects that I've seen come out in the literature, like the one relating to the I think you say fucosylation, the afucosylation of antibodies being in men. And then the other thing that I wondered if you wanted to comment on in the context of the EBV viremia is with age, you get that concept of memory inflation with sort of CMV or EBV responses. I've often thought, I wondered how much of those previous things are driving certainly some exaggerated hyperinflammation in the older age groups.
Yeah. So the EBV association to multiple diseases are now kind of coming out, and particularly to the multiple sclerosis, that link is really strengthened by a recent couple of papers. And similarly, I think that EBV may be associated with some forms of ME/CFS and other diseases. And these recent papers also implicate EBV reactivation as, perhaps being associated with long COVID. And so, and many of us actually have EBV. It's not just the EBV will, cause these diseases, but it may increase your chances of getting it. It's a very difficult thing to untangle, but we're actually looking at EBV viremia in the long COVID patients to see if that's something that's occurring in people even with mild disease. How the EBV induces long COVID or other diseases is still unclear. There's a very nice paper in Nature recently that showed that EBV antigen, the EBNA1, induces antibodies that are cross-reacting to glial cell adhesion molecule. And so there may be molecular mimicry that's going on with EBV antigens, and that might be relating to some of the autoimmune type of outcomes from that infection. But I think there's still a lot of work to do to try to make that link for other diseases.
Thanks. I think we'll just, we've got 10 minutes. I think we'll move across to some of the questions coming through in the long COVID. There was a lot of interest, and I thought it was really fascinating data on the distal inflammation mouse model that you had. The one question there were two questions. The one on the chat was around which variant you were using, and on your sort of a technical question on ACE2 expression. And so I guess that's related to the mouse model. And my question related to that was, did all of the mice get the same outcomes, or were there some that didn't get any distal inflammation?
Okay, so the strain that we used is the ancestral strain, the Washington strain, to infect. We are currently conducting a similar response with variants and even other kinds of respiratory infections like the influenza virus. And whether all the mice develop, so there's a range, obviously, of loss of oligodendrocytes, as I mentioned, is about 1/3 drop in the number that we can see, as well as demyelination. And it's not that all the mice lose all of it. That would be terrible. But there's a range. If you look at the paper, you can see the range of these responses, which I should have included in the presentation today. But there is a significant drop in the number of these oligodendrocytes, and significant increase in the reactive microglia, and significant drop in the neuroneogenesis in the hippocampus. So there is a range. And we also, I forgot to mention that we also used multiple strains of mice to demonstrate the same thing. So it's not a strain specific thing. We're now currently trying to understand whether it's specific to different variants or different types of virus even.
Great. And a related question to that, I guess, is have you started to look for what could be mediating this distal inflammation in terms of a blood marker or something? What is driving it?
Right. So that's a very important question. We're trying to understand how an acute respiratory only infection might be mediating such a chronic change in the CNS. And one of the, I guess the clues are coming from the fact that within this serum and in the cerebrospinal fluid of these animals, we can see elevation of various chemokines and cytokines, even at 7 weeks post-infection. And one of them is CCL11 or eotaxin 1, which had been implicated in other previous studies with a link to neurocognitive deficit. And so we're trying to study the molecular link between what's happening locally within the CNS as well as distally in the lung, and what's mediating this chronic change. And one of the other things that we're trying to do is if there may be an epigenetic reprogramming of the cells within the CNS, and we're trying to understand, what type of changes that are occurring in which cell types in the CNS that may be causing this chronic sort of state of activation in there.
Great. I see as somebody, the person that asked that technical question was also probing further about, was there any, how much human ACE expression did the mice have? And then were any of the responses related to that ACE expression? Like, was it dependent to some extent on how much virus was binding?
Yeah, that's a good question. So we did inject the AAV vector intratracheally, and we can see in our original paper, we described that there are multiple cells that are become transduced and are capable of expressing ACE2. So epithelial cells for sure, but some of the cell types within the lung and some potentially leukocytes as well there. So it's possible that it depends on the type as well as the extent of ACE2 expression that's occurring in the lung. We haven't dissected what cell types must be infected in order to create this effect, but that's a good question.
Great. Then there was another question that came through here about primary immunodeficiency. So, I think there's been quite a lot of literature about different cohorts and their connection with severe disease or even long COVID. What are your thoughts on patients with antibody deficiencies or T-cell deficiencies in terms of their risk factor for severe disease, their responses to vaccines, those kinds of things?
Yeah, absolutely. So we had a study in a couple of case reports of patients who are quite immunodeficient as a result of organ transplantation or leukemia. And in those settings, these patients were unable to clear the SARS-CoV-2 for an extended time period. And they really relied on things like monoclonal antibodies. And at that time, Paxlovid wasn't available. But anyway, I think those other types of measures will be necessary to protect these patients. And in particular, like these immunodeficient people are unable to mount a robust immune response with the vaccine, and they really do need sort of extra help with passive antibodies and so on. And in one of these cases, we saw that the monoclonal antibodies were quite capable of completely clearing the virus in that patient, whereas remdesivir was not. And remdesivir, in fact, we saw resistance develop after remdesivir treatment. So it really shows that, you need multiple kind of immune help in these patients, and a sort of readily available monoclonal antibodies are key. And we also have a study that we've done in the mouse model where we have a setting of B-cell deficiencies or T-cell deficiencies. And what we saw was that there is definitely an interplay between the T-cells and B-cells, but, deficiency in one of them can be compensated for the other. But if you lack both B and T cells, then the animals were completely unable to clear the virus. And so they become chronic carriers, sort of what we are seeing in these immunodeficient people. So yeah, we do need multiple therapeutic approaches to try to treat these patients as soon as possible.
Perhaps we've got time for one more question. And I think this is a question that a lot of people have been asking in the context of long COVID. What are your thoughts on vaccination? I know there's been some data that says it's good. Other people have got anecdotal stories that it's bad. So I don't know what your views from an immunological point of view based on the current hypotheses we have.
Yeah, so if I may break down that question to two parts. One is like, if you're vaccinated, what is the risk of developing long COVID?
Yeah.
And that appears to be about half. It's sort of the study, each study has different outcomes, but on average it's about half risk for developing long COVID. But we don't know if this risk reduction holds true for all the other variants, right? We don't know what happens with Omicron, for example. The second part of the question would be, is there a benefit of giving vaccines to treat long COVID patients? And that's also a mixed answer. People in general, like about 40 to 60% of long haulers feel better, at least transiently after vaccination, but that doesn't last in everybody. And so we're currently studying the impact of vaccination in long haulers before and after the vaccine to try to understand what changes promote this improvement in symptoms and how can we emulate this with a better treatment.
Great. I think that's, I know there are still quite a few questions there, but I think we've run out of time. It is 4 o'clock now. So once again, I'll just indicate a really great thanks. It was a fantastic talk, and your lab has really done amazing work in driving what we understand about this disease in such a short time. So big congratulations to you and your team. And thank you very much for taking the time to come and talk to us all, this international audience. And then, yeah, please, for the rest of the audience, stay tuned to the website. We've got a webinar coming on also related to COVID next soon by a presenter, Shane Crotty. So please join us for that. Otherwise, wherever you are, keep well, and thanks for listening.
Thank you.
Goodbye.
Goodbye.